In the intricate tapestry of nature, the concept of altruism weaves its way through various species, promoting cooperation for collective benefits. From the diligent bees to the vigilant meerkats, and even microscopic bacteria, altruism is a driving force that fosters survival and resilience. In human societies, altruism has played a pivotal role in fostering the communities we see today and has shaped the very fabric of human civilization. But what about the world of human cells, specifically cancer cells? Traditionally perceived as selfish competitors in the relentless race for survival, cancer cells present an unexpected twist. Our study dives into the unexplored territory of altruism within breast cancer cells. Cancer is often viewed as a cell-autonomous process, driven by genetic mutations, protein overexpression, and epigenetic rewiring. However, emerging evidence unravels cooperative societies within tumors that influence cancer processes, like therapy resistance. Our exploration began with the observation of elevated expression of a small RNA known as microRNA 125B in a subset of breast cancer cells. We conducted experiments to assess the impact of elevated microRNA 125B expression on our breast cancer cells. Using a co-culture approach, we combined breast cancer cells with different levels of microRNA 125B and exposed the mixture to the chemotherapeutic agent taxane, anticipating that cells with high microRNA 125B expression would survive the chemotherapy and dominate the culture. Conversely, we expected cells with low microRNA 125B levels to become extinct from the population, mirroring a survival of the fittest scenario. Contrary to expectations, cells with low microRNA 125B expression not only survived chemotherapy, but dominated the population. This unexpected outcome led us to hypothesize altruistic behavior, where high microRNA 125B cells might assist their low microRNA 125B counterparts, even at the cost of their own survival. We collected empirical evidence from different in vitro and in vivo models, and fitting our data into a social classification table like this, we show that the behavior exhibited by our breast cancer cells is altruistic in nature. There are many examples of altruism across the tree of life. And in each of these examples here, the types of altruistic benefit can be identified, such as indole production and sharing by the altruist in E. coli bacteria, or alarm call by the altruistic Belding's ground squirrel to alert the others of sightings of predator. So what is the fitness benefits provided by the altruistic breast cancer cells? We demonstrated that the altruistic breast cancer cells secrete protein factors such as CCL28 and IGF-BP2 to induce increased tolerance in neighboring cells to taxane chemotherapy. Apart from fitness benefits provided to others, altruism is also characterized by fitness disadvantage directed at self. And in these examples, the fitness disadvantage to self can be identified, such as growth retardation in altruistic bacteria, or increased risk of starvation in the altruistic vampire bats. So, what is the fitness disadvantage for the altruistic breast cancer cells? We show that growth retardation occurs in the microRNA 125B high altruistic cancer cells, and this is due to microRNA 125B targeting key regulators of cell cycle, such as CDK2, E2F3, and cyclin A2. We showed that microRNA 125B can bind to the messenger RNA of these cell cycle regulators, thereby leading to reduced expression of their proteins. With the expression of these proteins reduced significantly, cell cycle arrest then happens, leading to growth retardation in the altruistic cells with high expression of microRNA 125B. Interestingly, one upstream signaling event mediated by microRNA 125B led to two simultaneous events collectively underpinning altruistic behavior. Our study unveiled the activation of the NF-kappa B signaling pathway by high microRNA 125B expression. We further showed that different parts of this pathway contribute to either fitness disadvantage or benefits, playing a pivotal role in the dichotomization of events that define cancer cell altruism. As we unravel the mechanisms underlying altruism within breast tumours, a significant question emerges. Since altruism, with its fitness disadvantage, appears to be a losing strategy, how do altruistic individuals reproduce and persist in a population of selfish, winning strategies? Charles Darwin had noted this problem with the persistence of altruism in the light of the survival of the fittest paradigm more than 100 years ago. Drawing parallels with Charles Darwin's ponderings on altruism more than a century ago, we sought to understand the dynamics of altruistic breast cancer cells. Our experiments revealed that a purified population of microRNA 125B low non-altruists could give rise to brighter green microRNA 125B high altruists in a matter of days. Our findings suggest that the altruistic phenotype may be regulated by epigenetic rather than genetic means, as confirmed by modeling dynamics using evolutionary game theory. This insight highlights the potential for rapid interconversion of cells from one type to another, a mechanism observed in social organisms like honeybees and carpenter ants. Further investigation demonstrated the involvement of transcriptional factors, KLF2, and a histone acetyltransferase, PCAF, in the epigenetic regulation of altruistic behavior. The obliteration of KLF2 from breast cancer cells halted acetylation of histones, leading to the cessation of microRNA 125B expression and the end of altruistic behavior. But, with this understanding of why altruism doesn't become extinct in the tumor population, comes the question of preventing altruistic overpopulation within tumors. We have seen before that when a breast cancer cell has a higher level of microRNA 125B, it will secrete the trophic factors CCL28 and IGF-BP2. But we further discovered that these factors not only induce greater tolerance of the recipient cells to chemotherapy, but also downregulate their microRNA 125B expression level so that they will stay as non-altruist. This mechanism, known as lateral inhibition, involves intercellular communication and an intricate signaling circuit that allow adjacent cells to take up different social fates. This mechanism, together with the epigenetic mechanism involving KLF2 and PCAF, helps to ensure that the microRNA 125B high altruists persist as a constantly low-level and sparse subpopulation within the tumor. In conclusion, our study unveiled the mechanisms governing the manifestation, regeneration, and spatial organization of the altruistic subpopulation within breast tumors. So, why does this matter? Understanding cancer cell altruism offers crucial insights into tumor evolution and drug response, depicting tumors as dynamic social systems governed by cooperation principles found in nature. This perspective may also contribute to a broader understanding of cooperation and competition in driving the evolution of complex social systems in nature, such as the ant and the bees, that had fascinated Charles Darwin and many other natural scientists for over a century.